Hi everyone, it's me again, and we're going to finish up the material for the first exam, and then we're actually going to move on a little bit to cholesterol and phospholipids, which will be on the second exam, but there are some questions on the online quiz number 5 that relate to them, so I want to get through that as well. We were talking last time about lipids, and specifically we finished off talking about fats. And if you remember, this is what a fat looks like. It's the structure of a fat. And some of the characteristics that we went over with fats are shown right here. They are almost all hydrocarbon. They're almost all carbon and hydrogen. And so they are made up of nearly all nonpolar covalent bonds. They don't like water, they're hydrophobic, and they don't dissolve in water. The function of fats is to provide high energy, but in a storage form. So we store fat, which we don't really like, but it's there to provide us with an energy source if we need it. In fact, gram for gram, fats are more than twice as 2.5 times as caloric as carbohydrates and proteins are. So they're very high energy. We also use fats for insulation, for either protection, to protect some of our internal organs, or also for warmth. And we also said that the way that we make a fat is to take one molecule of glycerol, which is a 3-carbon molecule shown here, and we add to each of the carbons a long chain, very, very hydrophobic, nonpolar hydrocarbon. And so it's called a fatty acid, and it's attached to the glycerol by dehydration synthesis. And each one of these 3 fatty acids is attached by that procedure, and then you end up with a fat. So 1 molecule of glycerol and 3 fatty acids make up a fat. Now, I also said that fats are the only one of the 4 types of biomolecules that don't form polymers. So you're not going to have this fat here attached to another fat, attached to another fat, and so on. It doesn't form polymers, but they are made up of their own building blocks. As you can see, each one of these fatty acids is a molecule. It's considered a monomer attached to this monomer, and they add up to form a molecule. But you don't then make a whole bunch of these attached, so they don't then form polymers. So that's one difference from the other 3 that we talked about: carbohydrates, proteins, and nucleic acids. And then the other big difference is that they don't dissolve in water, whereas the other 3 do. And we finished off talking about that there were 2 types of general classes of fats. The 2 major types of fats are the saturated fats and the unsaturated fats, and that's where we finished off. So what's the difference between the 2? In a saturated fat and an unsaturated fat only differ in their fatty acid chains. And so if you go back here, these again are the fatty acid tails right there. And the difference between a saturated and unsaturated fat is whether there is any double bonds between the carbon atoms in any of the fatty acids. Okay? If you have no double bonds at all anywhere, and so they all look like this, all of the carbon atoms here, 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 here are all single bonded to each other, and all 3 of them are like that, that would make a saturated fat because it is saturated with the maximum amount of hydrogen. Remember, carbon binds to 4, can make up to 4 covalent bonds. It makes 4 covalent bonds, and in this case, if it's saturated, then it's going to have hydrogen on both sides because the other 2 bonds are to the next carbons. And so that would be no double bonds, saturated with hydrogen. However, if you have a double bond between carbon atoms, like this one right here, then those 2 carbon atoms, 2 of the bonds are taken up by the double bond. So each of the carbons can only now bind to 1 hydrogen atom. And that's why you only have 1 hydrogen for each of these and 2 for the rest. And so it's not saturated with the maximum amount of hydrogen, which would be 2 per carbon. So that would be called an unsaturated fat. Basically, the difference is whether you have any double bonds or no double bonds between those carbon atoms. So saturated fats, no double bonds between any of the carbon in the fatty acid chain, so it's saturated with the maximum amount of hydrogen. Unsaturated fats, one or more double bonds between those carbons in the fatty acid chains. And what we showed you before was what we call a mono unsaturated fat, because there's only one double bond in all of the fatty acids. So if you only have one double bond in all of the fatty acids combined, then it's called a mono unsaturated fat. If you have more than one double bond, in any of the fatty acids, even in combinations, then it's called a polyunsaturated fat, more than one double bond. Okay, so we have the saturated fats, and then we have two different types of unsaturated fats that I've talked about so far. And how do we describe them? This is the difference between a saturated fat and an unsaturated fat side by side. They come from different internet sources, so they don't look exactly the same. I could not find for the life of me them looking the same. But you can see that a saturated fat is linear in its fatty acids, completely linear across, and it's got all hydrogen around all of the carbons, no double bonds. These two are saturated fatty acids, but because this one is unsaturated, it makes the whole fat unsaturated. And that double bond there, not only does it mean that it can't have as much hydrogen, but it has a kink in it. And so it kind of goes kinky like that, okay? Kinky, whatever. And so it's not flat across like the saturated fat. And that makes a big difference in how they react at room temperature. And that is how their properties are, whether they are solid or liquid. And so another difference between the saturated and unsaturated fats is their appearance at room temperature. Saturated fats are solid at room temperature. And that's because those molecules have fatty acids that are linear all across. And so if you have a whole bunch of the fats together, they can pack really tightly because nothing's pushing them apart. They're all linear across. And so when things can pack tightly, they generally form a solid at room temperature. On the other hand, the unsaturated fat, because of that kink there, if you have a whole bunch of these molecules, like in vegetable oil, they're all the same, they are more spread out because the kink in this fatty acid pushes away the one next to it. And then that one pushes away the one next to it. And while molecules are further apart at room temperature, they tend to form liquids. And so butter, for example, is a saturated fat, and it forms a solid, packs tightly at room temperature, but vegetable oil is unsaturated. The molecules are spread apart more, and it forms a liquid at room temperature. So that's the difference between the two. Another difference between the two is where they come from or who makes them. In general, saturated fats are mainly from animal sources, whereas unsaturated fats are mainly from plant sources. Now, there are exceptions. So, for example, saturated fats include butter from animals, meat, beef from animals, and unsaturated fats, all of the oils that we have, vegetable oil, canola oil, olives, avocados, all of those contain unsaturated fats. But salmon, which isn't from an animal source, also is a really good source of unsaturated fats. And there are a couple of plant sources that are saturated, like coconut oil or palm oil, which are semi-solid at room temperature. But for the most part, saturated fats are mainly animal sources and unsaturated fats are mainly plant sources. And then a third difference between the two is how good they are for you, whether they contribute to heart disease and clogging of arteries or not. And in general, saturated fats, when they are elevated in your diet, you should keep them as low as possible because they will contribute to heart disease and will contribute to something called atherosclerosis, which is clogging of the arteries. And so you want to keep the saturated fats in your diet as pretty low as possible. You do need a little bit of saturated fat, but you want to keep it low because elevated amounts will contribute to these problems. On the other hand, unsaturated fats doesn't seem to have those problems. There is no association, even in moderate amounts, with heart disease and no association with atherosclerosis. And I wrote here except, which we'll talk about in a moment. But for the most part, unsaturated fats like vegetable oil, olives, and nuts and things like that, you want to keep those in moderate, good amounts in your diet. You want to don't take too much of them because of their caloric content. All fats have 2.5 times as many calories gram for gram as proteins and carbohydrates. But you want to keep a good amount of unsaturated fats in your diet because they're not associated with heart disease or atherosclerosis, and there does seem to be a protective component with them. They actually seem to protect against heart disease, and we'll talk about that in a moment. Now, you see this except here. And that's because there is a type of unsaturated fat, not the ones that we've talked about so far, not these from salmon or nuts or olives or avocados or vegetable oil or olive oil. Those are all good fats, and you want to keep those in a healthy range. You want to have that in your diet. But there is an unsaturated fat out there that is not good, that actually does contribute to heart disease and atherosclerosis even more than saturated fats. So that's where this except is. And what fats am I talking about? It's a type of unsaturated fat called trans fats. And most of you have probably heard of trans fats. If you haven't, then here you have it. They are bad. They're bad news. Okay. Now, trans fats are a type of unsaturated fat, and they are actually not made by plants. So they do not come from plants at all. Plants do not make trans fats, but some animal products, that is, some dairy products and some beef products, actually, meat products, actually have small amounts of these trans fats naturally. Okay, so that's a type of unsaturated fat that's not found in plants but are found in very small amounts in animal sources. And it's usually not the big problem, the animal source of trans fats that we're talking about when we're talking about the big problem with trans fats. Okay, the big problem with trans fats come from the fact that the majority of the trans fats that people get in their diet are artificial. That is, they come from a synthetic source. They are made in the laboratory. So they're artificial and they're found in many processed foods. So cake and cookies and microwave popcorn, if it's got hydrogenated oil added to it, and chips and things like that are often culprits of having sometimes significant amounts of trans fats, although they are decreasing them. They are putting limitations on them, thankfully. And the first example where trans fats became news was in margarine. So how many of you have ever heard of margarine? Margarine's been around forever. In fact, most people think, when you think of when margarine was made, people say, oh, maybe the 50s or the 40s or whatever. No, it was actually made, margarine was developed in 1869 in France, actually. There was a contest that because butter, which tastes so good, is pretty expensive. And not only is it expensive, but it's not very stable sitting out at room temperature. It starts to get rancid. And so there was a contest out there to try to get a substitute for butter that was lower in cost and actually was more stable at room temperatures or even warmer temperatures and didn't go rancid. And so what they did was they took vegetable oil, which was unsaturated, and they tried to make it partially saturated by adding hydrogen. So what you do in the laboratory is you take the oil and with a machine you actually throw hydrogen gas at it. And those hydrogens are hitting the fats and sometimes breaking the double bonds. And if you break the double bond, then that double bond becomes a single bond, and those two carbon atoms that had the double bond now become saturated. And of course, that's not going to happen to all of the vegetable molecules. Some of them are going to become saturated. Some of them are going to stay unsaturated. So you end up having a partially unsaturated vegetable oil. And it becomes a semi-solid. So the way that you can get something become more saturated from the unsaturated form is throw hydrogen at it, add hydrogen. And that's exactly what they do in the laboratory. So what they do is they take this unsaturated fat, and let's say this is vegetable oil, and now all of the molecules are going to look like this. Say there's 1,000 of these molecules in vegetable oil, and you compare that with 1,000 molecules of butter, which are all saturated. And what you're going to do is you're going to take these 1,000 molecules of unsaturated fat, and you're going to start throwing hydrogen at them. And some of the molecules are going to have their double bonds, that double bond broken, and hydrogen is going to add to it. And those molecules that have them will become saturated in a pool of unsaturated. So it's a mixture of saturated and unsaturated fats. And so we call it partially unsaturated. And it's partially hydrogenated. So it's not fully hydrogenated, because then the fat would be completely saturated and it would be essentially butter. But it's no longer completely unsaturated. It's a mixture of the two. And depending on how many of the molecules become hydrogenated, added hydrogen, then it will become more or less fully saturated. And then what will happen to this formerly completely unsaturated fat is it will become a semi-solid. It'll be easy to spread, which is really cool because you can't really spread vegetable oil on your toast. And it's called margarine. And it is actually stable at warmer temperatures. It doesn't go rancid. It's cheaper because it came from vegetable oil, which is much cheaper than butter. And whoever did that, I forget who it was, won the contest. And actually, they started adding coloring to it because it's actually a white color. So they made it look more like butter, and it doesn't quite taste exactly like butter, but it's good enough, cheaper, all that great. And not only that, but in the 60s and the 70s, they realized when they started looking at fats that saturated fats were bad, unsaturated fats were good, and they thought this was still partially unsaturated, so it's much better for you than saturated fats, and so everything was hunky-dory. Everybody was great, perfect. And it wasn't that way. Margarine was like the thing to buy in the 60s and 70s. I know it because my mom used to buy it all the time. The problem is they found out a couple of decades ago that it wasn't so good. And this is why. In the process of adding hydrogen here, some of these, because it's happening in the laboratory, some of these double bonds, even if they don't break, they shift from going to hydrogen on the same side to having one hydrogen on the other side. That's called an isomerization. Isomers, they look the same, but they're the same chemical structure, but the hydrogens are on opposite sides. And that is not natural in a vegetable oil. Vegetable oils come from plants, and plants only make fats that look like this, where if you look at that, and if you look at the original fat I showed you, if you look at this fat here, see how there's the double bond right there. See how the two hydrogens are on the same side of the carbon, on both sides, they're on the same side. That's called the cis configuration, CIS. Cis means same. And so that configuration is natural for plant unsaturated fats. That's the only thing that plants make when they make their unsaturated fats. They put them in that configuration. But in the laboratory, when you're adding and you're throwing hydrogen at this, sometimes that configuration can flip so that one hydrogen's on one side and one hydrogen's on the other, and that's called the trans configuration. And that's not natural for vegetable oils, and it's found in small amounts in animals, but it's not natural in plants. And it looks like this. So here is the natural form of a unsaturated fat. And of course, some of these fats are getting saturated by adding hydrogen in the laboratory. But the ones that aren't, some of them are isomerizing and flipping so that they're in the trans configuration. And that's what a trans fat is. When a cis fat flips in the laboratory. And that's not a good thing. Why is it not a good thing? Because trans fats are associated with an increased risk of heart disease. Not only that, but they're increased even more than saturated fats. These are the worst fats you can have. How bad are they? Well, the FDA, when they recommend how much saturated fat you should get in your diet, and I said it's a small amount compared to unsaturated fat. Well, how much trans fat should you get in your diet? According to the FDA, none. You should not be having trans fats in your diet. Now, it's impossible if you're eating animal products not to get any. You're going to get some trace because that's, some animal fats have a little bit of trans fat. But processed food, they recommend keeping that to zero. That's how bad they are. And in fact, if you look at a diagram here showing the relative risk of heart disease associated with increasing the intake of certain types of fat, if this is your baseline 1, where there's no risk difference, where you have an average diet of a normal amount of saturated and normal amount of unsaturated fats, and you have no trans fat, and it's like you're basically, it's a good diet, okay? And you have no risk, extra risk. If you add more saturated fats than normal, your risk goes up to 1.17. That means your risk is 17% increased over a normal healthy diet of heart disease. Now, if you elevate your monounsaturated fats over a normal person's with no risk, it actually decreases. As you can see here, it's down to 0.81 compared to 1. That means there's a 19% decrease in your risk of heart disease if you add more monounsaturated fats to your diet. And it's even better for polyunsaturated fats. It's down to 0.62. So there's a 38% decreased risk over a normal healthy diet if you add more polyunsaturated fats. But look at the trans fat. Trans fats are so bad, it's almost 100% increased in risk. It's a 93% increase in risk. It's almost twofold more higher increased risk for heart disease than a regular diet. That is really bad. And that's why they say not to have trans fats. Now, how do you know if you have trans fats in your diet? How do you know? I usually have in my face-to-face class, I ask somebody always has a bag of chips. So I say, okay, take out the bag of chips. And we start looking at the ingredients. And you look at the fat, and the bottom it'll say how much trans fat. But be aware that they try to fool you. That is, if it says 0 grams trans fat on your bag of chips, they're lying, probably, because they can get away, based on the FDA, with saying 0 grams fat, even if it's anything less than 0.5 grams per serving. So let's say you have 3 servings in that bag of chips, okay? and they're saying 0 grams trans fat. And who is going to have one serving of a bag of chips? One serving is like 3 chips. Okay, nobody has one serving. Everybody just eats the whole bag. And so you're eating the bag, and that's 3 servings. You could have up to 0.49999 grams per serving. Multiply that by 3, and you almost have 1.5 grams of trans fat just from the bag of chips. And you're already over your limit, which was 0 in the first place. So be aware. Now, well, if anything that says 0 grams fat is a lie, how do you know if it's, how can you find out if something has 0 grams fat? Well, first of all, eat things that are not processed, okay? Because look on the ingredients. Look for partially hydrogenated vegetable oil. That is a clue. If it says that, there's likely trans fats in there. Okay. Now they try to get rid of the trans fats. They can actually, it would cost money, but they can try to eliminate the actual trans fats from the regular cis fats in partially hydrogenated vegetable oil. But if it says that in the ingredients, chances are there are some trans fats in there, even though they're trying to get it down even lower and lower. And in fact, they've actually made a mandate that margarine has a smaller percentage than it used to have. And so most of the margarines will say that it has less than 0.5 grams, which in the past it might have had a couple grams. Okay. So you got to be really careful. But here's one way that you can also look for ways to keep your trans fat low. Not only look for the ingredients and make sure it doesn't say partially hydrogenated vegetable oil, but also if it says no trans fat, no means no, okay? 0 grams does not mean 0, but no means no. And there's the clue right there. Okay, they cannot say no trans fat unless it truly has 0 grams trans fat. And so you're not going to very much see that very often, but you will see 0 grams, which is not necessarily being truthful. Okay, so that is it for the first exam. Okay, we finished fats, and so all the way up through all the lectures I've done now, up till now, will be on the first midterm exam. However, there are a couple of questions. I think there's 3 questions on the online quiz number 5 that is a continuation of lipids. And so I want to continue those off. This will not be on the first exam, this material, but I'm going to include it in this video so you can actually get some of the, you can finish those answers, okay? You could probably figure them out yourself, but I'm just going to continue on in this lecture, and we're going to talk about the other lipids. So this second lipid that is important is completely different from fats. It looks completely different. It has completely different properties. The only thing that makes it similar to a fat is that it's also mainly a hydrocarbon, and that is cholesterol. So cholesterol, remember, this is not going to be on the first exam, but it is also an important lipid. And cholesterol is also almost all hydrocarbon. In fact, there's even more hydrocarbon content to cholesterol than there is to a fat. And it is so, because it's almost all hydrocarbon, it's very nonpolar. Nearly all of the bonds in this molecule are covalent nonpolar bonds, nonpolar covalent bonds, which means that they are hydrophobic. They don't like water. It doesn't dissolve in water, just like a fat. But take a look at the structure of cholesterol. It looks so much different than a fat. Okay? In fact, this is one molecule that is a molecule that stands by itself. It's not going to have other molecules attached together like with a fat through dehydration synthesis. Okay? It is just a molecule, and that's it. It's a big, bulky hydrocarbon, except for this OH. That's the only part that is polar, and it stands by itself. Okay? So that is cholesterol. Very bulky, has several several carbon rings, and we've seen that before when we looked at back when I looked at functional groups and I was looking at estrogen and testosterone. Well, they are also steroids, just like cholesterol. Cholesterol fits in a class of molecule called steroids. In fact, cholesterol is the most prevalent steroid in our body. Okay? And it has a number of important roles. So let's talk about some of these important roles. The first thing we need to talk about is that, yes, we do need cholesterol. People are like, oh, do we really need cholesterol? I hear that it's really, really bad. Well, no, we do need cholesterol. We do a lot of things with cholesterol in our body. However, our liver makes nearly and can make all of the cholesterol we need. So you don't have to get it through dietary sources. You should get it. You should get some through diet, but you don't literally have to because your liver can make all of the cholesterol it needs. In fact, in a normal person, the liver is making 80% of your cholesterol, and the other 20% you're getting from dietary. Okay, so when you have cholesterol in your blood, about 80% of that is made actually by your liver, and only 20% comes from your diet. And then you can actually lower your dietary amount. You really can't lower your amount that your liver makes unless you take drugs. Okay, but you can lower your dietary amount, but then your liver will upregulate to get the amount it needs. Okay, but the answer here is no, we don't need cholesterol in our diet, but we do need cholesterol in our bodies for some very important functions. So let's look at some of those functions. So one of the important functions of cholesterol is that it is a component of our plasma membrane. Okay, so plasma membranes, as we'll see when we talk about phospholipids, are made of lipids. And there's embedded things in the plasma membrane or the cell membrane, but it's made of lipids. And one of the components of the plasma membrane in animals is cholesterol. So what is it doing there? Well, here is your plasma membrane. And so we're going to see that the plasma membrane is mainly this type of lipid shown here with the little gray things. Those are called phospholipids, which is our next topic. But here is the cholesterol. So wedged between these types of lipids called phospholipids are molecules of cholesterol. Okay. Now they're not very prevalent compared to the other lipids, the other phospholipids, but they are wedged in there periodically. So what are they doing in our plasma membranes, in our cell membranes? What they're doing is regulating how fluid this membrane is. The plasma membrane is not static. Static would mean just staying there like this. It's not like a solid. Plasma membrane is actually a fluid. It actually moves. The components, the molecules in the plasma membrane are moving back and forth all the time. They're actually moving pretty rapidly. So it's sort of like a fluid. And cholesterol helps regulate that fluidity. Okay? You don't want that fluidity to be too slow, which might happen if the cells get too cold. So if your temperature goes down too much, like if you experience hypothermia, you're outside and it's freezing and your body temperature starts going, your cells start getting colder. You don't want your cell membranes, you don't want your plasma membranes to freeze because there's important things in there that need to move around. And so in that case, what cholesterol will do is speed up the fluidity of the membrane, make those molecules go faster. So that's what cholesterol will do. If the membrane gets too cold, cholesterol speeds up those molecules to prevent them from freezing. If the membrane gets too warm, if you have a fever and things are going too fast, because the warmer things are, the faster molecules go, then cholesterol slows them down to get them back to homeostasis. So cholesterol's job is to speed up or slow down the membrane components and change the fluidity to get it back to normal. That's how it works in our membranes. It also has another role that has nothing to do with our plasma membranes and has everything to do with our digestion. So, how many of you have ever heard of something called bile? You might have heard of bile when you're like, bile. Oh, God, I'm like tasting my bile. Well, you're not really tasting your bile. When you puke, if you're puking your guts out and you just keep puking and puking, and then all of a sudden you're dry heaving and nothing's coming out except this kind of yellowish green fluid, that's bile. Okay. Bile is made in your liver and it is made from cholesterol. So cholesterol function is actually to be a component of this substance called bile. Bile is actually made from cholesterol and salts like sodium, chloride, things like that. Salts that are very polar and cholesterol, which is very nonpolar. And what it does is it acts like sort of like a detergent in your digestive system. Acts like a detergent because when you eat fatty foods, remember fats don't like water, and your saliva and your digestive solvent is mainly water. So when you eat dietary fats and it goes down into your stomach and then starts going into your intestines where the digestion is going to take place, those fats form these fat droplets to get away from the water solvent of the digestive fluid. And they are not accessible to digestive enzymes as fat droplets. They're kind of masked. And so the digestive enzymes can't get in to digest those fats. And so what bile does is it helps to break up the fats into individual fat molecules. And so then the digestive enzymes can come in and chew up the fat. And so it acts like a detergent to dissolve or break up those fats. It's not digesting the fats, it's just breaking up the fat droplets so that the fat digesting enzymes can get access. That's what bile does. Okay, sort of like what a detergent does in your sink to the fat droplet. It dissolves those fat droplets. Okay, so again, bile is made from cholesterol and some salts. It helps to dissolve and make the fats accessible to digestive enzymes in your intestine. Where is bile made? Bile is made actually in an organ called the liver, which is one of our biggest organs. And the liver then makes the bile from cholesterol and salts and then stores it in a little outpocketing of the liver called the gallbladder. And that's where it's stored and it's ready to go. So then when you eat a fatty meal, all the gallbladder has to do is contract, squirt out some bile, and the fat as it comes down will start getting dissolved and broken up, and then the enzymes can get it. And so the gallbladder, as I mentioned, is this little outpocketing of the liver, and it looks like this. So you're not going to have to memorize this or draw any of this, but this is part of your digestive system. Here, right here, is your food tube, also known as your esophagus. And as food's coming down, it enters the stomach and starts to get broken up by acids. But your fats don't even get started to be digested at all there. In fact, they're forming fat droplets. All the fats that you've eaten are forming droplets to get away from the watery solution in the stomach. And then the fats come out of the stomach and enter the beginning of the small intestine, right here. And that's called the duodenum. And that is where all the digestion is going to take place, where fats are going to get digested, where carbohydrates are going to get digested, where proteins are going to get digested, down to their monomers so they can be absorbed into your bloodstream. But they won't get digested unless they are accessible to the digestive enzymes. And most of those digestive enzymes come from this organ called the pancreas. That's where most of the digestive enzymes are made, and then they are sent into the duodenum. Now, one of those digestive enzymes, the one that's going to break down fats, is called lipase. Okay, it breaks down lipids. So the pancreas is going to send the lipases into the beginning of the small intestine, waiting for the fats to come down. But those fat droplets, unless they've been broken up and dissolved, the lipases can't get at them. And that's the job of bile. So bile, which is made in the liver from cholesterol and salts, is stored in this little pocket, outpocketing of the liver called the gallbladder. And when you eat a fatty food, okay, the gallbladder contracts and squirts a little bit of bile into the beginning of the small intestine. And along with the lipases, then the bile can dissolve the fats, break them up, and then the lipases can actually digest them into fatty acids and glycerol that they were made from. And then they can get into the bloodstream and you can use them by your cells. Okay? Now, that's basically what's happening. But can you live without your gallbladder? Do you have to have your gallbladder? And the answer is no. You don't need your gallbladder. In fact, gallbladder surgery to remove the gallbladder is a pretty common surgery. You do need your liver because it's not just bile that's being made by your liver. Your liver is doing a lot of other really important things, but you don't need your gallbladder. And so if your gallbladder is removed, what will happen is your liver will still make bile, but it will have to be made on command. There's no place to store it anymore. And so the liver can make the bile and it can come out of this little duct here, but it's not going to be stored and ready to go. So you're not going to be able to release as much when you have a fatty food. So that means that fatty foods are not going to be as dissolved as well. They won't be digested as well. And most of them will be eliminated, just sort of like cellulose and like lactose if you're lactose intolerant. And so that's a problem. So they say that if you've had your gallbladder removed, you should avoid fatty foods. Because if you don't, you're going to end up pretty much running to the bathroom a lot with greasy stools because your fats are going to be eliminated. It's not a pretty sight. Believe me, I know that because my mom had gallbladder surgery. And then she kept calling me up and saying, Marilyn, why do I have to go and run to the restroom all the time? I don't understand it. And I said, well, are you eating fatty foods? And she goes, well, yeah, my doctor said I could eat whatever I wanted, the same diet. No, her doctor should have told her she shouldn't eat fatty foods. So I told her that, and I don't know if she's changed, but anyway, I don't live near her, so that's okay. Um, anyway, okay. So the third role of cholesterol is our final role, and that is to make other molecules. And so besides making bile, cholesterol is also used to make vitamin D, which is an important vitamin that's made from cholesterol, and it's also activated by sunlight. So you can make all of the vitamin D you need from your cholesterol, but then it can be activated by sunlight. So you can make even more. So most people, if they live in Southern California, are not deficient in vitamin D. But the other thing it also is involved in making, which is really important, is all of the steroid hormones. Remember we said cholesterol is a steroid. Well, it's important in making all of our steroid hormones. We don't make very many. We only make about 5, but a couple of them are really critical for reproduction. And so those reproductive hormones, also known as the sex hormones, estrogen or estradiol and testosterone, remember they look a little bit like cholesterol. They are the same ring-like structure, and they are made from cholesterol. And so here is estradiol, here is testosterone, and it looks very similar to that cholesterol that I showed you before, like that. There's some differences, but it looks a lot like it. And so they're all called steroids, and both of these come from cholesterol. Now, testosterone. I need to talk a little bit about this one because, yes, it comes from cholesterol, as does estradiol or estrogen, same thing. But there is one of the roles of testosterone, besides giving males secondary characteristics and allowing sperm production and all that stuff, is also to build muscle. And so testosterone is critical in building uh, the bigger muscles that males have compared to females of the same size. And some people will take synthetic forms of testosterone that mimic testosterone to get bigger muscles, and those are called anabolic steroids. Anabolic meaning build up steroids, and they build up muscles. They mimic testosterone. So here's your testosterone. Here are some of the synthetic anabolic steroids that are on the black market, so to speak, that you shouldn't be taking. You can take them if you prescribe them for certain reasons, but it's very rare. It's actually illegal, except under the most extreme circumstances, and is absolutely illegal if you're in any kind of competitive sports because it gives you a competitive edge. Plus, they are dangerous because they have their own side effects, but they all are based on testosterone, as you can see there. And so anybody who knows anything about sports probably knows this guy right here, Barry Bonds, who was on steroids, okay, and he finally admitted to it. He actually is the current home run king, and currently there's probably nobody even close to him. The only active player that is within range, which probably will never get even close, is Pujols, which is he's on the Angels, but he's not going to make it, and everybody else falls short. He's got about, I think, 762 home runs, and so he's the home run king. But his record is tainted because he took steroids, and you can tell he did by looking at pictures of him and comparing him to what he looked like when he started his career. And so when he started his career, he was with a different team, the Pittsburgh Pirates, but look at the difference. That's what he looked like when he started. He was scrawny, and then he started taking steroids, and boom. Okay. Now, steroids do anabolic steroids like testosterone will actually help you generate muscle. And so the way it works is as follows. You will not need to know this now. We'll be doing this later when we talk about hormones and signal transduction, but I'm just going to show it to you now because it's really interesting. So when you take testosterone, it goes through your bloodstream and then it actually enters your cells, and there's a receptor in cells that are targets for testosterone. Only certain cells have this receptor. And so testosterone is only going to work on certain target cells. And then it binds to the receptors, and then the whole thing, the receptor with testosterone, enters the nucleus of your cell and binds to specific genes and turns those genes on and makes those genes generate proteins. And in a muscle cell, the proteins that are generated are the contractile proteins that we'll talk about later on. So if you take testosterone, it's going to affect your muscles by allowing your muscles to make more of these contractile proteins. The whole muscle cell will grow. Okay, it won't divide. You won't get more muscle cells, but the actual muscle cell will grow because there's more proteins in there, and those more proteins are contractile, so there'll be more of a force that those cells can produce, and you'll be able to lift more weight and you'll be stronger. And so there's actually an experiment that was shown on fibers of muscle fibers from the thigh, for example. Each one of these are muscle cells. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9 muscle cells before treatment, and they're all a certain size. After treatment with testosterone, you still have 9 cells. So muscle cells do not divide, but what they do is get more of the proteins inside these little tiny fibers, and those fibers grow, and then the whole muscle cell grows. And since, again, it has more of those contractile proteins, it can create a higher force, and you get stronger legs in this case. So that's muscle cells. And so that, again, brings us back to cholesterol being used to make testosterone and then taking anabolic steroids. What are the dangers of taking anabolic steroids? There are a lot of dangers, okay? One is it will put stress on your heart. And so people that take anabolic steroids run the risk of heart attacks and strokes. Another, because you can get blood clots. Another risk is that individuals that take steroids, and it doesn't only have to be testosterone, but steroids in general, sometimes medications, can get something called roid rage or steroid rage. And what that is violent mood swings that can actually be violent. And so people that have known to commit murders and domestic violence when they're on steroids. Another thing is that when you take anabolic steroids like this, you downregulate your own levels of your own natural sex hormones. So for guys that are taking anabolic steroids that mimic testosterone, they will actually put out less of their own testosterone, and they will generally potentially become infertile. Not only that, but their testicles shrink to like grapes. No, raisins. Raisins. Okay, so if you really want to, not take steroids, listen up, guys. You don't want your testicles to be shrunken like that. Females and males, both, if they take anabolic steroids, can go into that kind of puberty phase where you're getting acne all the time. So not only on their face, but on their back. Males, because the enzymes, because both estradiol and testosterone both come from cholesterol, and the enzymes are can go back and forth to generate these things. If you have too much of this, you start making estradiol. So males will start actually developing breasts, and females will start, because they have too much testosterone, if they're taking anabolic steroids, will start growing facial hair, and their voice will deepen. So these are all bad side effects that you don't want to have. And that's basically all due to taking anabolic steroids. But again, how it relates to cholesterol, cholesterol is the precursor for steroids in general. Okay, we'll talk more about these and steroids when we talk about the endocrine system later on. Okay, I'm going to finish up the last part on phospholipids, and this is actually going to be a precursor to our next topic, which is the plasma membrane. Now, phospholipids, they are different from the other two that we just talked about, because phospholipids actually have a nonpolar part, but they also have a very polar part, a small polar charged part to them, which the other lipids, fats, and cholesterol do not. What a phospholipid looks like, it looks sort of like a fat. It has glycerol, it has fatty acids, but it only has 2 fatty acids. And in the third position, instead of a third fatty acid, it has a modified phosphate group. Remember what a phosphate group is? We saw that in nucleotides. And if you remember what the phosphate group looks like, it's negatively charged. So it's polar. And so it's very different from the non-polar hydrocarbon we saw with the fatty acid. Very, very different. And so what a phospholipid consists of is glycerol, 2 fatty acids, and then this modified negatively charged polar phosphate group. And so the fatty acids in phospholipid are going to be coming off of glycerol. Remember, glycerol is a 3-carbon molecule, and then you have your 3 fatty acids in a fat. Well, the fatty acids in a phospholipid will go down, but then the third position where you normally have a fatty acid, That phosphate group does not like the nonpolar fatty acids. It hates them because the phosphate group's polar. So it flips over to the other side and generates this weird looking molecule that looks like this. So there's your glycerol, the 3 carbons. There's your 2 fatty acid chains, which we call hydrophobic tails. And there's your polar phosphate group, which is flipped over to the other side because it doesn't like to be near the hydrophobic part, because this is hydrophilic. And so this is the structure of a phospholipid. And this part is the polar phosphate group, which is negatively charged. And so a simpler way to draw it would look like this. There are the fatty acid tails. This one happens to be unsaturated. This one happens to be saturated. There's that glycerol. There's that phosphate group. And it has something attached to it, but we don't care about that. This is polar. These are non-polar. We call this the polar head or the hydrophilic head of a phospholipid. We call these the hydrophobic or non-polar tails. And in general, nobody, this is the way it would look in a book, but it's hard to draw that all the time, so we generally draw an abbreviation like this. This is a phospholipid. There's the polar head. There are the nonpolar tails. This is hydrophilic. This is hydrophobic. And to actually make it a little bit more complete, I'm actually going to put the glycerol in there just for kicks, like a bow tie. And then, of course, you know me, I got to put a happy face on it. And just to show you that this is negatively charged, we're going to put a little negatively charged halo on it. But from now on, you're just going to see it like this. That's going to be our phospholipid. So what do phospholipids do? They have one role. They have one role. And I'm going to show you what that role is by doing an experiment. What would happen if you threw a whole mess of phospholipids into water, into a bucket of water? You threw a whole bunch of phospholipids into water. What would happen? Now we're throwing these phospholipids and they're being forced to go into water. And then we have a third one who's scared. Okay, so they're going into the water. What's the easiest way for them to arrange? Well, I think the easiest way for them to arrange would be like buoys, okay, where the polar heads would go under the water because they're happy being their hydrophilic heads, and the hydrophobic tails would stick out away from the water. So it would look like that. Okay, there's that scared one. This one's angry for some reason. But what if there was no more room there and you were forced to go underneath? You could do a couple of things. One, you could actually put in the water all of your polar heads on the outside facing the water and squish in all your hydrophobic fatty acid tails in a single, like, small little ball and squeeze out so no water's in the inside. That could work. Or, check this out, you could make a phospholipid bilayer where the water is on one side and the other side, the polar heads are facing the water and the hydrophobic tails are facing each other. That's called a phospholipid bilayer. But what happens at the edges? If this were the glass beaker, everybody's happy in here, everybody's happy here, everybody's happy here, but once we get to the edge and we're up against glass, which is polar, by the way, the polar heads are happy, but the tails are up against the glass and they're not happy because that's nonpolar hydrophobic against glass. So what could you do to make everybody happy? What if you took, let's say all this is water, they're all in water. You took this phospholipid bilayer, and as you went across, instead of going straight across and hitting the walls, you made a circle, like all the way around, like this. So the polar heads are facing the water. The polar heads on the inside are facing the water all the way around, and the nonpolar tails are in the middle. And what if, fortuitously, you captured some DNA and some goodies? What would you have? You would have the first cells. And that is how it's believed that the first cells came to be. That is, a whole bunch of phospholipids were found in water, and they oriented themselves in their best way possible to get away so that the nonpolar tails could get away from water and the polar heads could face the water. And they just happened to capture some genetic material and enough goodies. And this is now your cytoplasm. This is your extracellular fluid. And that's how we believe the first cells evolved. And so the next topic is going to be the cell membrane and all of the different functions of this plasma membrane as it relates to biology. So I hope you enjoyed this. I'll talk to you later.